Alright, chemists, let's ferment some glucose to ethanol. This is pretty super sweet. Actually getting less sweet as we lose all the glucose. So, here we have glucose plus oxygen gives you yeast, water, and carbon dioxide. This is a very familiar reaction to you. This is the backwards photosynthesis. This is respiration. Aerobic means that there is O2. Anaerobic means no O2. But we'll get to that. Alright, so I want you to pause very quickly and balance this equation. Alright, now, because you're a winner, I'm assuming that you've already done this. Alright, so we put a 6 there, 6 there, 6 there, and all is good with the world. Now, it's important to realize, this will come into a second, this is an exothermic reaction, which means its enthalpy is a whopping minus 2,803 kilojoules, that's okay, per mole. Okay, so we're all good there. That's all very exciting. That's respiration. That's it. That is yeast using up its food, which is glucose. Now, then we have anaerobic respiration. When there is no oxygen present, the yeast can still survive. They're pretty tricky because it turns out that you'll die from not being able to get rid of the carbon dioxide, before you run out of oxygen. So the yeast are pretty tricky. They've got an enzyme in there called zymase, which breaks down sugar or glucose or fructose when there is no oxygen present. Okay, and it looks like this. Oh, hang on, we've done it again. There we go. They're all good. That molecule is correct. So you get glucose in the presence of zymase goes to ethanol and carbon dioxide. I would like you to please pause it and balance that equation. Alright, again, because we know that you're a winner, we know that you've done this, which means you put a 2 there, and you put a 2 there. And if you couldn't get it, whatever, we'll get there. It's tricky. So, let's have a quick look at this one though, and this is relevant. This is why the yeast would rather do this reaction than this reaction, because the point of respiration is to get energy from your food. So delta H in this one is minus 74. Check media sheet. 0.4. 74.4 kilojoules per mole. Alright, sweet as. So that's all nice and straightforward. Now, the living conditions. Okay, yeast cells they use as a catalyst, so they need to be alive while this is happening. So it must be performed at the right temperature, otherwise the yeast dies. He's like, above 30 degrees, I'm going to die. Below 30 degrees, I'm probably not going to do much at all. Alcohol is also toxic to the yeast, which is another reason that it's not ideal, aside from the low level of energy. So if there's too much alcohol in there, it'll die. So if it's above 15%, the yeast will kick the bucket, and we've got to get rid of it. So we have to take the alcohol away. And we do this with a distillation. Oh no, we remove it, the mixture, and then we distill it after that. Alright, so fermentation. Basically, fermentation is a biochemical reaction in which sugars are converted to, or broken down into, ethanol and carbon dioxide. Now, under the action of an enzyme. It's a really complex series of reactions, which means this to here is missing a bunch of steps. There's a lot more in there, and we're not going to worry about it at this point in time. So the reaction is exothermic, which means it's going to build up heat. So we need to take the heat out. Now, so this is because they're producing energy as they go. So we get CO2 and ethanol happening right up until we run out of sugar, and that's all the conditions reach a fatal level for the yeast. All right, so let's have a look at the process. This is not a short process. There's quite a bit. Maybe now it's time to take a break and come back to it. Okay, so you crush and grind the organic biomass. In this case, it is to get out cellulose. Alright, we hydrolyze it with dilute sulfuric acids at about 100 degrees Celsius for 2 hours, and this breaks the cellulose into its monomer units of glucose. We filter out the remaining stuff, essentially the solid, and we redo it again with the acid to just sort of nail it in there and keep it nice and to get the rest of the sugar out there that we can. So we hydrolyze it to get more sugar, or as much as we can. The solid that's this step here that I just said. So this creates a little loop that brings us back here, and it starts again. Then we add calcium hydroxide to the solution. So what that does is that neutralizes it. Okay, so that's really important if we're going to put it in with a bunch of yeast. So we go calcium hydroxide, which is this, plus the H2SO4, which is in the solution there, gives us calcium sulfate plus 2 waters. And that's a neutral solution. Good thing about calcium sulfate is it's insoluble. Okay, so if we wanted to, we could put a little S there. You need to know these equations to explain it. Because it's solid, we can filter it out. And then we just ferment the remaining sugar that's in that solution using yeast. And this gives us carbon dioxide and ethanol. We distill it to separate the CO2, the ethanol, and that's very energy intensive. And the solid waste can be dried, and this is actually really interesting. The solid waste you've got left over, you can burn it, and this gives you heat or electricity for the plant. So it's kind of keeping as much of it in-house as possible. Alright, this is what it looks like as a flow chart. Although, yeah, okay, so fine. So you've got your organic biomass. You crush and grind it, you then hydrolyze it. You add, in the hydrolyzation, that should be shaped like that, by the way. For this, hydrolyze it, you're adding the acid, you then filter it out. The filtrate goes off, sugar's in acidic solution, blah, blah, blah. You add more acid to the solid residue, which is then hydrolyzed, and then goes back into this, which is then filtered out again here, and you get rid of your solid residue. Which then you get sugar solution, which you ferment, and it gives you CO2, an ethanol mixture, which is distilled, and this ethanol mixture will be about 12 to 14%. You distill it, you never let it get 15 because it kills it, and you've got ethanol and byproducts and waste. Now, we can summarize that, so be cool. Basically, you start with your organic biomass, which is then hydrolyzed, gives you a sugar solution. The sugar solution is fermented. You get an ethanol mixture plus CO2. The ethanol mixture is distilled, and you get ethanol. And that's it. Well done for hanging in there. I'll see you later.